Namaskar for all the viewers. Welcome to the world of microbes. I am Dr. Nagbhushan, Assistant Professor, Department of Zoology, VSK University, Ballari, Karnataka. Today we shall discuss upon general microbiology. When we think of microbiology, the very first question arises in mind: what is microbiology? It is the study of microorganisms. For the first time, Robert Hooke designed his own microscope to observe living things. His contemporary Dutch Anthony Philip van Leeuwenhoek also designed the microscope using spherical lenses, which had a magnification of 50 to 300 times fold. He had even written over 200 letters to Royal Society of London about his findings and discoveries. He made microscopic observation of the tooth scrapings of the oral cavity and called them as animalcules. You can see it in that picture. In the history, if we go back to 17th century, there were various theories proposed in support of origin of life and evolution. Many believed in the theory of spontaneous genesis, that is, abiogenesis. Later, experiments of Italian physician Francisco Redi were very decisive enough in leading to the abandonment of spontaneous genesis doctrine. Italian naturalist Lazzaro Spallanzani was the first to provide evidence that microbes did not arise spontaneously in organic infusions. At the same time, French scientist Louis Pasteur also used swan-necked flasks to disprove abiogenesis. John Tyndall also proved that optically empty air contained absolutely no microbes as he passed a beam of light through it in a box experiment. There are n number of important discoveries in the field of biology, especially when we come into microbiology. Some of the milestone contributions in the field of microbiology dates back from 1796 by Edward Jenner. He realized the process of immunization, which we call it as vaccination. Louis Pasteur discovered rabies is caused by organisms other than bacterium. He also devised various methods to immunize anthracis and fall cholera diseases. Machnikov showed cellular theory of immunity in that there is one theory known as humoral theory, which says that soluble substances present in the body fluids are responsible for showing immunity. Behring said that toxins produced by pathogenic bacteria are neutralized by antitoxins produced in the blood serum of immunized organisms. Twort showed that bacteria are also subject to viral infection. Whenever bacteria is infected by a virus, we call that virus as a bacteriophage. The term bacteriophage was coined by D. Herrell. Wendell Stanley isolated, purified, and crystallized one type of virus which causes infection in the plants, TMV, tobacco mosaic virus. Hershey and Chase discovered that genetic material containing nucleic acids, it may be either DNA or RNA or both, penetrate the host cells during infection. Sabin and Tam indicated that HSV, herpes simplex virus, belonging to the family herpes, may cause cancer in human beings. Very recently, Prusiner in 1997 coined the term prions, where viruses are made up of only proteinaceous chemical compositions. In order to understand microbiology in detail, there are various tools and techniques involved. The comprehension of the things that are being studied is facilitated by various numbers of the tools. The commonest kind of the tool that is required for microbiological understandings are the microscopes. Microscopes are classified into two categories. The first category microscopes are called optical microscopes or simply light microscopes. Second category of microscopes are electron microscopes. The power of magnification in this type of tool, microscopy, ranges from 50 to 2 lakh times fold. Optical or light microscopes make use of the qualitative light and variable focal length of the lenses, whereas electron microscope makes use of electromagnetic lenses and an electron beam to obtain the magnification. There are different kinds of optical microscopes listed in this picture. You can just see one is bright field microscopy, dark field microscopy, ultraviolet microscopy, fluorescence microscopy, phase contrast microscopy, and so on. Electron microscopes are of two types: transmission electron microscope and scanning electron microscope. The main function of these microscopes is not only meant for magnification after observation, but they are also meant for micropreparation techniques and preparing specimens for the observation too. When we understand the process of mounting technique using the microscope, we come across with certain terminology. A couple of terminologies have been discussed here in detail. Resolution power. It is the diameter of the smallest object or the least distance present between the object and the lens system. That means, if you take human eyes into consideration, it can be defined as the ability to separate two dots placed at 0.25 mm by human eye, whereby two dots placed in less than 0.25 mm apart cannot be distinguished by human eyes. That means if we place two dots and the gap between two dots is less than 0.25 mm, we will not be able to distinguish that there are two dots. It appears as if it is made up of a one dot or a single line. Same thing is applicable in the magnification under this terminology, resolution power. In abbreviation, RP depends upon the wavelength of the light admitting lens, which is determined by NA, numerical aperture. Resolution power can be expressed by the expression RP is equal to intensity of the light, that is wavelength of the light, divided by 2 into numerical aperture. The shortest wavelength of visible light will give maximum resolution of the object. That means smallest distance that can be resolved, that is, finer will be the details revealed under the microscope. For example, if the wavelength of yellow light is 580 nanometers, therefore RP is equal to 580 divided by 2 into 1. That equals 290, where 1 in the denominator is the numerical aperture value of the lens. That means when you see this light of yellow, 87,000 lines are visible to an inch. Numerical aperture is the refractive index of the medium multiplied by sine, that is angular aperture, divided by 2, that equals n sine theta. Therefore, greater the numerical aperture, better will be the resolution power. That is why we make use of immersion oil when we want to magnify the compound microscope images to about 1000 times or 2000 times, where refractive index of the oil is 1.5 other than 1. In order to mount the specimens, there are various mounting techniques involved. We group the mounting techniques into two types. One is wet mount, other one is dry mounting. Wet mounting is one where the sample is suspended in the liquid. The specimens are suspended in the hanging drop method, that is called wet mount. The second one is a dry mount where the specimen to be observed is dried, fixed, and stained spheres. It all requires a method of staining. Staining is the nature of the dyes has organic compound containing both chromophore and oxochrome group linked to a benzene ring. The chromophore imparts color to the specimen, whereas the oxochrome imparts property of electrolytic dissociation. Stains may be categorized as acidic, basic, or even neutral. Various kinds of staining methods are also involved, such as simple staining, differential staining, gram staining, acid-fast staining, special staining for morphological studies, and so on. Other than microscopy, there are different kinds of instruments that may be useful in the field of microbiology. Some of them may be differential centrifugation. Before that, there will be microanalytic techniques required in order to mount the specimens in the electron microscope. There has to be disintegration of microorganisms, there has to be separation of subcellular structures. Only then we will be able to get the details of every smaller bit of the specimen to be mounted. Other instrumentations include differential centrifugation, gradient centrifugation, radiography, adsorption chromatography, column chromatography, thin layer chromatography, partition chromatography, gas liquid chromatography, ion exchange chromatography, electrophoresis, and so on, so on, so on. The microbes as such are our friend and foe too. No man is exempt from them. These microbes are microscopic to ultra microscopic in nature, in the sense they are so tiny that we have to make use of the microscope in order to see them. Some of the organisms belonging to bacteria and viruses are so tiny in nanometers range that ordinary magnification of the compound microscopes won't be able to get the details of it. Hence, we need to make use of electron microscopes to get the details of them. Such organisms are called ultramicroscopic. These microbes are found everywhere. They are ubiquitous in distribution. In the sense, we know that all living organisms exist in three spheres of the biosphere. One is atmosphere, hydrosphere, and lithosphere. In the extremes, we don't find any. Ubiquitous distribution in the sense the organisms that are capable of showing its habitation, existence in all possible habitats. Some of the microbes are obligatory organisms, some of the organisms are facultative parasites. Some microbes do require oxygen for their survival. They are called aerobic organisms. Some do not need oxygen for their metabolic pathways, such as anaerobic microbes. Majority of the microbes are harmful and some of the microbes are useful, in fact. The microbes can be classified into 5 categories. They fall under various kingdoms too. We find them in kingdom Monera, we find them in kingdom Protista, we find them in kingdom Mycota, we find them in kingdom Metaphyta, we also find them in kingdom Metazoa, wherein protozoa can be placed in kingdom Protista or in Animalia. If you go in detail, the microbes are of 5 kinds. One is the virus, viruses, bacterium, bacteria, fungus, fungi, protozoa, protozoans, alga, algae are singular and plural terms. Kingdom Monera includes viruses, bacteria, and cyanobacteria. They are prokaryotic organisms without having a definite nuclear membrane. Whereas fungi, protozoans, and algae do have well defined nuclear membrane, nuclear envelopes, hence they belong to eukaryotes. So, microbes are both prokaryotes as well as eukaryotes. There are various bases taken into consideration for the purpose of classification of these microbes. Viruses can be classified based on the chemical composition they bear. Know one more thing, that viruses are showing the properties of both living as well as non-living matter. They are the simplest living creatures, having made up of only two chemical compositions. One is the protein coat called capsid with capsomeres, sometimes enzymes within, and the core is made up of nucleic acid, either deoxyribonucleic acid or ribose nucleic acid. So based on the nucleic acid core, the viruses can be classified into DNA viruses and RNA viruses. They can also be classified based on the host range. They may be also classified based on the presence or absence of the envelopes. Those who have virulent nature and non-virulent nature could be another way of classification. There are various other methods of classification of virus exist today. Bacteria can also be classified based on their shapes, such as cocci, bacillus, we have spirillum, we have comma shape, and so on. Based on the nutrition, bacteria can also be classified. Based on staining response, also the bacteria can be classified as gram positive and gram negative. Fungi can also be classified based on the number of cells they produce and based on the reproduction types they exhibit. Protozoans are classified based on the number of cells they bear, unicellular and multicellular, chlorophyllous or non-chlorophyllous, so as the algae. In this picture, you can see various kinds of the microbes. Starting from left top corner, we have different kinds of viruses having the protein coat in the form of the extensions called crown or corona. Then we have got the TMV, tobacco mosaic virus, to the right hand top corner. Below we have got that bacteriophage infecting the host bacterium. Then we have got coccus bacterium found below, right hand bottom most corner. Then we have got bacilli. To the left hand corner you find both, all the three, coccus, bacillus, spirillum. Above that we have the comma shaped bacteria. Various kinds of bacteria are observed here. In this slide, we can see different kinds of protozoans. There are various protozoa made up of a single cell. Others may be present in the form of the colony. To the right side, we have got agaricus, the mushroom, the yeast, the rhizopus, the fungi infecting the food molds, the bread molds, and so on. These are different kinds of the algae, chiefly composed of chlorophyll types A, B, or C. Some may not even have the chlorophyll. Different kinds of diseases are seen in both plants and animals caused by the viruses. If we take human beings into account, we are familiar with various common viral diseases exhibited in human beings. It may be HIV, poliomyelitis, common cold, herpes, mumps, diarrhea, Ebola, rubella, influenza, yellow fever, brain fever, meningitis, rabies, measles, hepatitis, MERS, SARS, COVID, and so on. Where do we find these microbes is another question. As we said, they are universally distributed, ubiquitous in nature. Even in the extremes also you find some of them. They are found in cold and freezing altitudes of even less than 45 degrees Celsius, minus 45 degrees Celsius. They are found to be seen in the hot springs in the bottom of the ocean, where the temperature shoots up to 250 to 350 degrees Celsius, where we find some of these microbes existing. They are found even in the deserts, hot deserts, even in the marshy places where methanogen kinds of the microbes do present in large numbers. Of course, they are found in the free places, they are found in the free living form, and even as the hosts, either being present on the surface of the hosts or being present as endoparasites. Initially, we said that these microbes are both friends as well as foe. If you consider these microbes are friends, how friendly are they? It dates back to our ancient time, where there was ancient biotechnology and ancient microbiology, where the microbiology started from our kitchen. Our forefathers, grandmothers, and grandfathers knew the best utilization of these microbes. In the kitchen, we are familiar with the preparation of the curds, preparation of idli, dosa, cakes. All these are being produced as the food cuisines. Without microscopic organisms, without these microscopes, we will not be able to produce this duff and the batter. So we make use of the good microbes out here in order to prepare curds, idli, dosa, and so on. So consumption of such kinds of the food products, which are having good number of harmless bacteria, useful bacteria, can act as probiotics. They have a capacity to even prevent human health from being exposed to some of the commonest illnesses. Other kinds of bacteria are responsible for enriching the fertility of the soil. Such are called nitrogen fixating bacteria. They help in fixation of atmospheric nitrogen as nitrates in the soil, which enhances the productivity of the food crops. In the modern biotechnology or microbiology, we have known the process of utilization of the microbes and their byproducts for the betterment of human race and the environment. Large-scale production of enzymes, hormones, pharmaceutical products, alcohol, steroids, secondary metabolites, antibiotics have taken birth in the recent past, and we continue to utilize the microscopic organisms byproducts for the betterment of human welfare. Vaccines are being produced that are used as antibodies in order to activate our active as well as passive immunity, thanks to the discoveries made by Louis Pasteur, Alexander Fleming, as well as Edward Jenner. How harmful are these bacteria, the viruses, the fungi, the algae, and protozoans? Thousands of diseases are actually caused by these microbes in all kinds of organisms, starting from bacteria, plants, animals, even human beings. We cannot list them out here in a short span of time. But in order to study the entire disease aspects, we have a terminology called as disease etiology, which involves these terms: pathogen, the organism which causes illness in an organism; the invader which disturbs the normal metabolic pathway is a pathogen. Pathogenicity stands for the degree of infection caused by a pathogen in the host, the ability of the pathogen to induce improper physiology in the hosts. The pathogenicity is dependent upon the chemical nature of the invading pathogens, invading microbes. It is understood that plastic is having the least pathogenicity, whereas the proteins are having the very high degree of pathogenicity. In the sense, any microbe which is having a protein is found to be causing illness. It will have high degree of pathogenicity, and the infection will be of greater degrees. Infection is the mode of transfer of the pathogen from the infected animals to a healthy animal. The mode of transfer could be communicable or non-communicable. In the sense, the organisms that come under communicable diseases do get transferred through certain media. They may be airborne, waterborne, it spreads through the water, foodborne, sexually transmittable diseases, through the vectors, or even vague, unable to discover exactly the mode of transfer. Some of the pathogens require carrier for their transfer from one of the organism to other host organisms, passing through the vectors. We call them as the carriers. Without the carrier, their life cycle is incomplete. The diseases can be classified into two types, communicable diseases and non-communicable diseases. If there is a physiological malfunctioning and it results in the disease, it is said to be a non-communicable disease. The vice versa is said to be communicable disease. We are focusing upon communicable diseases caused by the microbes. We need to understand the cause of the disease. What are the modes through which the pathogen might have entered into the host and so on. What are the clinical symptoms that are exhibited by the host need to be studied in order to diagnose the actual disease shown by the organism. Its effects on the body need to be studied. And on the diagnosis of the disease, we have to give the therapeutic treatment, or in some cases, prophylactic treatment. Prophylaxis is one where treatment is given before the onset of the disease. We are familiar with three terminologies. One is epidemic, endemic, and pandemic. Epidemic is the disease spread only in the local people, in a limited number of individuals. Endemic takes up the spread over a slightly increased geographical area where large number of individuals exist. Pandemic is one where the spread is very rampant, going to the states and nations and the world, which we are facing right now. Hence, microbes are both our friends as well as foe too. Nurture the friendly microbes because they need to survive to let us survive. Thank you very much for this opportunity. Thank you so much.